Hello, Bio30, and welcome to Spermatogenesis and Oogenesis, also known as Gametogenesis, meaning both gametes rather than splitting into two. Normally, we would talk about this conversation around how sperm and eggs are made in chapter 14 and 15, but since it fits nicely into our next unit as well, we're going to bring it up now and talk about it. So in your notes, you are going to go through the process of spermatogenesis. So sperm, meaning like what males produce, so the male sex cell. But if you look at spermato is like the beginning parts of all the words we're going to talk about, and genesis is the formation of, so the creation of, okay? So we're going to start with a cell. Now that you guys have seen mitosis and meiosis, you now know that cells have chromosomes in them. And especially in meiosis, you know that these are going to be some cells from father and some cells from a mother, so male and female. We're going to start off by calling this structure a spermatogonium. Spermatogonium is the first name of the cell that's inside that seminiferous tubule that's going to develop. As it goes through, it's going to then produce a primary spermatocyte. So what you're seeing here is the inside of this developing sperm, okay? It's going to replicate, cross over, and then it's going to split into two as a secondary spermatocyte, okay? So here we have a full set of chromosomes. So this, okay, is the start of meiosis, and then it splits down into half. So we call that the division part of meiosis, right? So your spermatocyte has a full set of chromosomes, then we split it into two to become a secondary spermatocyte. These secondary spermatocytes then break into four. So this is now the equal part, the equatorial part, or equational part. I always say that word wrong. But anyways, it's the equal part of division. We had two chromosomes here. We now have two in every type. So here we had four, we go down to two. Here we have two, now we have two across the board, okay? So this is the spermatogonium, the original, the spermatocyte as it's developing, primary, secondary, and then we call these spermatids. These spermatids are inside the seminiferous tubule and they get sent up to the epididymis to mature. So now these are the very first stages of sperm, then they go into the epididymis and they mature into full-blown sperm. So this is the process of spermatogenesis. So you need to write it down, show what each stage is called, and understand which parts are undergoing the first stage of meiosis and then the second stage of meiosis. So here's meiosis 1 and here's meiosis 2. And the reason I say that is as we get into talking about mutations, I'm going to be referring to things at anaphase of meiosis 1 and anaphase of meiosis 2. So let's just make sure we label again. This is meiosis 1, this is meiosis 2. So again, just to reiterate, this is happening inside the seminiferous tubules, which are inside the testes. So inside the testes, right here, you have all these tubule structures. And remember, it's a tube. So it's a round part with a hole through the center because the sperm, as they develop from the outside in. So they develop from the outer side of the tube into the center. And once in the center, they can travel through the tube, up through here, and into this section here, which is known as the epididymis. The epididymis allows them to mature, and when they're ready to leave, they go up and they leave through the vas deferens. So this is a cross-section of a seminiferous tubule under a microscope, and you can see here are our cells that are what we would call spermatogonium. And then it develops inwards. You can almost see like these stringy-like structures here. Those are the tails of the spermatid. All these little dots are all spermatids lining the inside of the tube. So if we were to cartoon this, it would look like this. Here are my spermatogonium. Here are my primary spermatocytes. Here are my secondary spermatocytes. And here are my spermatid. All right? So that is the first step. Now we're going to talk about oogenesis. So oogenesis is now the development of an egg in a female reproductive. So what we have here is the oogonium. The oogonium is just representing of the ovum, or what we're developing as a follicle. Again, it has 4 chromosomes in it, and then they replicate. All right. So now, again, just like in spermatogenesis, we have 2 sets of, or a tetrad, okay? Crossing over has occurred. We call this the synapse, right, when they come together. And then this is called the primary oocyte, so the start of the developing follicle. Okay, now this developing follicle will split into the secondary oocyte, because remember, we're going through primary meiosis, meiosis 1 right here, right? We're going down to half the amount of chromosomes. But unlike sperm, eggs do not develop 2 eggs. We develop what we call an oocyte, the secondary oocyte, and something called a polar body. Females only develop or should only develop 1 egg through every cycle. So our body as females makes sure that they don't develop 2 by creating something called a polar body. And what a polar body is essentially all of our energy goes into developing 1 very, very healthy egg, and this one is not. So it doesn't have anything left in it, no nutrients, and it essentially it dies. It doesn't develop into anything. So as we go through the process of oogenesis, rather than both of these dividing, only the secondary oocyte divides. So again, this is meiosis 1. Now we're going through meiosis 2. We're going from 2 chromosomes down to 2 chromosomes. But much like before, we only want 1 egg, and we only want 1 really healthy egg. So rather than putting all of its energy into dividing into 2, it puts all its energy dividing into 1, and it creates another polar body. So this is the ovum, which develops the egg, which is released during ovulation. So again, the process is very similar to spermatogenesis, except this time ooo, meaning egg, and genesis to create an egg. So the oogonium is our initial developing follicle. As it matures, it makes a primary oocyte, a secondary oocyte with a polar body. Your secondary oocyte splits into an ovum and a polar body. This part here is meiosis 2. This part here is meiosis 1. So remember, ladies and gentlemen, this is happening inside the follicle, inside the ovary. So as it is passing, okay, so over here, this is ovulation. You can see the releasing of an ovulated oocyte. These are the developing follicles, okay? So here's my primary follicles, my secondary follicles, okay? So your primary follicles start with an oogonium, then we develop into primary oocytes, secondary oocytes as it travels through, and then we release our ovum, okay? So in the world of oogenesis, it's this part of the cycle. It's the part that develops the egg. It has nothing to do with the corpus luteum. Okay, it's only developing this ovulated oocyte right here. So get both of those into your notes, and if you have any questions, please let me know.